Yeah. All right. So without getting too propeller head on me, can you talk a little bit about the engineering that enables this? Because there've been it seems like for years there's been discussion around outside the liver, right?
Yeah.
So can you share with us just a little bit on sort of the fundamentals, I guess, of that?
Yeah, in a very high level way. So the technology that we're based on comes from Caltech. There's a woman named Viviana Bradinaru who has a lab in Caltech. Two of the people in her lab, Nick Flintanis and Nick Godin, together we call them the Nicks. So I may refer to them as the Nicks during this call. The three of them are the scientific co-founders. And what they did was they take that virus and manipulate the amino acid sequence on the surface of the capsid. And that creates literally billions of variants. By doing that, manipulation, it used directed evolution to create billions of capsid variants.
Mm-hmm.
And then what we have built is a screening process using robotics and that automation that can filter and screen, all the different NHP tissues. The important part is we're doing all our work in NHPs and in human cells, not just in mice. And so we go from the billions of capsids down to thousands of capsids, down to dozens of capsids, down to the one that has the criteria we're looking for each disease. So for every disease, you set out in advance almost like a target product profile, like you would for, let's say, a clinical stage product. Here we create what's called the target capsid profile and say, okay, if we want to get to Parkinson's, here's the cells we need to impact. Here's the brain regions we need to get to. And so we set up that criteria in advance, and that filtering process, screening process, allows us to get down and identify that capsid.
Mm-hmm.
So hopefully that wasn't too rudder head.
No offense to the propeller head.
Yeah, propeller head.
No, that's great.
But hopefully that explains the secret sauce. And so that is our platform. But that platform has been productive, and now that platform has created assets that are on the cusp of going to the clinic and helping to treat patients that have our target diseases.
Yeah. Then that process, is it a very time-consuming process?
It started, it's almost like, I'm not in the software world, but, they say that once you have your first software, every successive version goes quicker. The turnaround time is quicker. It's the same sort of thing. So the first time we did it took a little longer, and every single time that process has shrunk down now to the point where it's, in the weeks and not months.
Yeah. That's a ML AI enabled process, I'm assuming?
No. No, it's mostly robotics and automation enabled.
Yeah.
For us, certainly AI and all of that has great promise in our industry. But for what we're doing biological screening, we think is the best way to get the end result that we're looking for versus modeling it or, AI in silico approaches.
Yeah. So we talked a little bit about the macroeconomic environment in 22. And I mean, if you're able to pull through that, you've got a leg to stand on. There's a foundation. And then in this field of genetic medicine, there is a microeconomic environment. And on any given day, don't ask me what investors are thinking, the partner you're looking for. But you've got to differentiate, right? You've got to create something that keeps genetic medicine in the, therapeutic du jour. Tell me about that. Like what's that been for you? And what levers and buttons do you have control over to navigate that?
Yeah, I think everybody understands the potential of gene therapies and genetic medicines more broadly. And we've seen some of those successes, but we've also seen a lot of failures. And so I do think genetic medicines in some ways have fallen out of favor in the marketplace. And people are certainly excited about things like obesity and immunology. But we exist to solve the challenges that led to this falling out of favor. Mm-hmm. So I think what's fallen out of favor is some of those generation 1 therapies that are using, AAV9 and can't be delivered IV without accumulating the liver, or you have to do direct brain injection. And so all the things that have made genetic medicines at least in our space and gene therapy, fall out of favor, is why we exist, to solve those challenges. So sometimes it's a little frustrating that we get swept up in, that broad view. But the important part is that the progress that we're making, and it's tangible progress, I think will certainly help continue to differentiate us out of the pack of the group that has kind of been a little bit laggards. But I also hope we're going to be at the forefront or the tip of the spear of helping bring a new set of enthusiasm and a new era of enthusiasm to what we're doing.
Yeah. I get the sense that some of the enthusiasm around cell and gene therapies, both cell and gene therapies, some of that enthusiasm gets quelled by the complexity of manufacturing, supply chain, last mile supply chain, if you will, and delivery to patients. Big challenges there. How does Capsida approach or address some of those kind of back end challenges?
That's a super important point. So in addition to what I was describing about our differentiation around capsid engineering and getting to where you want to go and not going to where you don't want to go, one of the other challenges that has set back a lot of gene therapies is quality of manufacturing. So thanks to our investors, again, Westlake and Versant, who understood that and were willing to make an investment in our own manufacturing facility. So we have a 15,000 square foot manufacturing facility that's in our same building where our research labs are in Thousand Oaks. And so we have complete control over timelines, cost, quality through that manufacturing facility. And on top of that, we build in manufacturing screening room, that screening process that I talked about going from the billions down to the one. Not only are we looking in that screening process for capsids that meet the profile that we're looking for, but we're also looking for capsids that are manufacturable, at least in as much quantity and scale and ease as AAV9. So that's one of the early screens that we do to help us select the capsid, and that wouldn't be possible if we were using a CDMO or if we didn't have that capability. We would, and I'm sure there's great CDMOs out there, no criticism, but you lose control. And anytime you do a tech transfer, you introduce risks, and you can't have the screening as early in the process as you would like, and you don't have that collaboration of being in the same building between the research team and the manufacturing team. For all those reasons, we made the decision to invest in that facility, and it's been a wonderful decision. And not only has it enabled our programs, but quite frankly, we're enabling our partners' programs. So I mentioned the partnerships that we have with AbbVie and Lilly. We also have a partnership with CRISPR and Sally. But we just got an option, as an example, opt-in for one of the lead programs from the AbbVie collaboration in neurodegeneration. We are going to be the manufacturer for that program. So that's great validation. But not only did that building that facility enable our programs, but it's enabling the AbbVie program to go forward. We have a relationship with a sister portfolio company in the Westlake and Versant portfolio called Therapeutics. And they don't have a manufacturing facility, and we do. So we created a relationship in 2023 where we would be a manufacturer for them. Novartis bought them. And now they're a Novartis company, and they're going to continue to use us as the manufacturer. So that capability has been gold for Capsida, but it's proving to be that for the AbbVie program and hopefully other programs from AbbVie, and for these now Novartis programs.
Yeah. Did that manufacturing facility come online under your watch, or did that? It was built. It came online just as I was joining.
Yeah. So I joined in January of 2022, and that's when it was coming online. But the decision to make that investment was made just prior to when about a year prior to when I came, and the great manufacturing team that we had built that out.
Our friends at Alston & Bird set us up with some amazing space to record during JPM week. The firm's national healthcare and life sciences practice has more than 100 attorneys actively involved in the healthcare industry across the full spectrum of legal disciplines. The signature strength of this practice is its ability to master complex representations that draw on the coordinated expertise of its regulatory, compliance, public policy, transactional, corporate governance, securities, FDA, biotech, IP, government investigations, and litigation practice areas. Alston & Bird represents life sciences companies and their partners in corporate stages, ranging from private to newly public to well-established, and in a variety of stages of product development from preclinical to post-approval commercial launch. Learn more at alston.com and tell them you learned about them on the Business of Biotech.
Do you have any insight into what kind of a heavy lift it took to convince investors that manufacturing in-house is the way to go?
It was not cheap. I certainly won't quote what it is that it costs, but I've certainly heard the discussions with our investors and with our team, and it was expensive, but it was worth it for all the reasons that I mentioned. It would be way more expensive if we had all the reasons that you even brought up. The question is, if we had these great programs and they weren't manufacturable in the right scale or quantity, and we had to then go to another CDMO and do a tech transfer, and then, a delay in our programs, or, hopefully, having any kind of not having any quality issues, or the FDA shutting you down because of your quality issues, would have been way more expensive than making that initial investment.
Yeah.
And we have, like I said, we have great investors that had enough wisdom and willingness to make that investment. I'm thankful that they did.
Yeah. The previous episode to this one was with Stefan Scher from 3T.
Yeah. So we have a common investor in Wesley.
Right. Yep. Yeah. He spent some time there, I believe, in fact. That discussion sort of centered around the shape shifting that, an R&D focused company moving into the clinic has to endure, or, the decision making that a CEO needs to make around personnel and where resources are allocated. Obviously, they are not manufacturing in-house, but I would imagine that would be a pretty big sort of shape shift, not only to do in the first place, but to maintain. So 3 years in now as CEO, tell me about how you're maintaining expertise, qualification, momentum in that manufacturing facility.
Yeah. Well, first of all, we have a great team that's super experienced. They've been making biologics for literally a lot of them, 30 years. Super experienced team. And they're committed to our cause and our mission. They're a really great team that a lot of them have worked together for a long time and kind of came over as a team. And they've been great for the rest of the organization as well in terms of culture. And so they're pretty self-motivated. But yes, of course, there's a few, pivots that you make. You kind of pivot from being a platform to having assets, from having assets to getting them into the clinic, which that's the stage we're at, and going from, let's say, on the manufacturing side, kind of vector production, small scale for animal experience, to then, expanding to suspension, broad-based manufacturing for humans, and it's different quality level of specifications. And so all those have been super exciting pivots that we've made successfully up until this point. And the next and most important pivot we're making is filing INDs, which we'll do here in the first half of the year, 2 INDs, one for our program in developmental and epileptic encephalopathy caused by a specific mutation, by the STXBP1 mutation, and then a Parkinson's program caused by GBA mutations. Both of those, knock on wood, will be submitted and approved INDs in the first half of the year, and hopefully we'll be treating our first patients in Q3 for both programs. So all of those have been exciting evolutions, next steps, that at the end of the day, one of the things that we've talked about is the great science that we have isn't just for science sake. We're not an academic institution. We created this science so that it helps in the service of patients.
Yeah.
And so that's also been kind of a North Star for all of us, whether it be the research team, manufacturing team, certainly the clinical team, why all of us are here.
Yeah.
And so it's a really exciting time for us making that big pivot.
Will that manufacturing facility support clinical trial supply.
GMP.
Yeah.
We just finished the GMP manufacturing for our first program, the STX BP1 program, and we're in the middle of finalizing our GMP for the Parkinson's program. And so that is at the FDA's quality specs. We've had pre-IND meetings where we've submitted, the data that we have, and we got, positive or supportive feedback from the FDA. But yeah, so this is a clinical and commercial grade manufacturing facility.
Yeah, I asked that question in part because some of the, so often when we think about genetic medicine, we think like rare disease, ultra rare disease.
Yeah.
Some of the indications that you guys are looking at are not so rare, certainly not ultra rare.
Yeah.
So it seems to me there's a different sort of capacity requirement potentially as you move the product through the clinic.
We certainly have the capacity to do the upcoming clinical trials. But as those programs progress and as our partners programs progress, I hope we'll be in a situation that we need to expand our facility. We have the capability to expand our facility in our current footprint. That would be a wonderful situation to be in.
Mm-hmm.
But yeah, so you were talking about like indications.
Yeah.
We purposely picked indications that would, well, first of all, that have an unmet need that we think we can help these patients. That's first and foremost. But we selected them to be kind of bookends. So to your point, in gene therapy, a lot of the traditional programs are in pediatrics, ultra rare or small diseases. So the STX BP1 program is certainly not ultra rare, but it is considered rare. It's about 5,000 patients in the US and Europe. But and in pediatric population. That's kind of the traditional place gene therapies have played. And on the other end, we selected Parkinson's GBA, adult population, neurodegeneration, bigger patient population, about 300,000 patients in the US and Europe. Mm-hmm. So we wanted to show the bookends of what's possible with our capsids, with our cargo optimization capabilities, everything. And so that's why we picked those two as the lead programs. And it also, to your point, showcases what's possible on the manufacturing side too, to be able to accommodate both of those adult patients that require more and broader populations, as well as, the rare condition in a younger patient population. So I think those indications, besides those programs, besides being best in class to help patients who are desperately suffering, also are a great showcase of what's possible through our technology.